Good evening, everybody. Gentlemen, take a seat. My name is Roger Kirby. I am currently the president. I have the honor to be the president of the Royal Society of Medicine, and you are all very welcome. Especially welcome because this is the 31st Ellison Cliffe Lecture. We had to have a virtual lecture last year. This is the first time we've actually had this many people sitting in these seats for a long time. So you're very welcome. I think we have 450 people online watching the famous Mark Lythgoe talking about imaging and advances in biomedical imaging. Just a little update on where we are at the Royal Society of Medicine. We've had a difficult pandemic, like I suppose most people in this room. We have a deficit of about £6 million, unfortunately. I'm looking at Ian Barmer there, and he'll know what a deficit of £6 million means for an organisation like this. Not good. But there has been a silver lining to the cloud of the pandemic here. We have, to use the in phrase, pivoted to digital. And some of you, or maybe quite a few of you, hopefully have seen some of our digital offerings, including COVID series. Somebody described that as a sort of cult series on a Thursday lunchtime at 12:30. We've had all sorts of famous docs on there, including Chris Whitty. Jeremy Hunt actually is appearing in 10 days' time. I'm going to talk about the investigation into the way that the pandemic's handled. That will be very interesting. And we've also run a series called In Conversation Live that started here in this actual auditorium with Stephen Fry. And when we had Stephen Fry here, we had people queuing down the road to see him. And that kind of gave us the idea that we would run a series called In Conversation, based really on the program In the Psychiatrist's Chair that some of the more elderly people like me in the room will remember. So just in conversation with various personalities, and we've had Andrew Lloyd Webber, we had Stephen Fry, we've had George Osborne, all sorts of famous people. So, and those are on Wednesday evenings at starting at 7 o'clock, run through till 8 o'clock. Well, next week we have David Spiegelhalter. You might know probably the UK's most famous expert on statistics from Cambridge University. He's here for the Dangle Lecture, and it's a free lecture, and there are free drinks afterwards a week today, so you might like to register for that if you're not already registered. I'd like you to tune in, and I imagine quite a few of you are members of the Royal Society of Medicine, but I'd like to remind you don't have to be a clinician to be a member of the Royal Society. In fact, we're trying to broaden the Royal Society of Medicine out to healthcare workers beyond clinicians, to managers and nurse specialists and physicians, assistants, etc., because medicine is teamwork these days. It isn't just about the doctor and the patient, it's the doctor and the team around him. So we do encourage you to join the Royal Society of Medicine and help us move things forward. We've been around for 215 years, the RSM, so we want to keep it going for at least another 215 years and beyond that. And in order to do that, we will need your help and assistance. So you're very welcome, and with no further ado, I'm going to introduce my previous president, bar one. Simon Wesley came in between, sort of interrupted B's transition to me, and did a great job, of course, and B himself did a fantastic job as president. So B. Sheffield is going to introduce our speaker, Mark, and I'll be back again to monitor the questions, moderate the questions. So do think of some questions as you're listening to this evening's talk. And welcome to everybody online. B.
Thank you, Roger, and welcome to all of you, as Roger has said. My job is to welcome Mark, and indeed to welcome your family, Mark, Helen and your children here tonight. It's a great pleasure to see you all here tonight, and I'm sure we're in for a great evening. But something about Mark, who started out with a master's in behavioral science and then a PhD in biophysics, subsequently from UCL. He's the founder and director of the UCL Center for Advanced Biomedical Imaging. Its pioneering capabilities are aligned to meet today's medical challenges. Mark has been awarded £45 million or thereabouts for his collaborative programme of imaging research, a fantastic amount of money and a fantastic achievement. And he's also Director of Biomedical Research at the Francis Crick Institute. He's published over 250 papers in high-impact journals, Nature, Nature Science, The Lancet, and so on, and has an H index of 55. Now, that actually means, if you take the work of George Hirsch, who started the Hirsch index, it means he's an outstanding scientist. If you get up to 60, he becomes a unique individual. And I think he's already a unique individual, having met him not so long ago. But additional to his pure scientific efforts, Mark is committed to public engagement in science. He's director of the Cheltenham Science Festival. I think that's probably getting to be even larger than the Cheltenham Racing Festival. And he won a prize in 2015, the Neuroscience Prize, the Prize for Public Understanding. He's been awarded Salford University's most successful and notable graduate. So he's really a hugely talented individual in many spheres. And I found out over lunch the other day, he's clearly a fine fisherman as well. But interesting, I was reading, and I'm sure many of you also read Cotswold Life, those of you that are resident, at least out of London, and he stated in 2014 that he was also the most useless man on the planet. I beg to disagree with that assessment. He's always said that science is incredibly exciting for me, and I think you're going to find that in tonight's lecture. So without further ado, Mark, can I invite you to take the stage? Mark Lythgoe.
Good evening, everyone. Thanks, Bea. Thanks, Roger, for the wonderful introduction. I can't tell you how excited I am to be here tonight to give this award lecture. I want to welcome you all here and also the people that are online with us tonight. I want to start by acknowledging the Percy and Caris, who endowed the Ellison Cliffe Award. They have a love of how physics could be applied to medicine. But it wasn't as simple as that. What they really appreciated was someone that could communicate concepts to a broad audience with real ease and simplicity, which puts a considerable amount of pressure on me this evening. And I hope that I can fulfill both of these loves, which is to bring medicine and physics together and also to be able to communicate that. I'm going to start with a very unconventional introduction to myself, something that I've only just started to talk about, something that I deeply regret and something that I'm not particularly proud of. I started this journey at 18 and I failed all my A-levels, and because of that I never went to university. Instead of that, my first job was here. This is in a plastic moulding firm in South Manchester, and I worked on line number 27, which is here. I'd take large bags of pellets and I'd drop them into this hopper. They'd be melted in this device here. They'd be heated up, and then a screw would push it out through a die into a particular shape. It would get pulled out, cooled off, and then chopped into lengths. And I would sit there once I'd set up the machine for 12 hours a day packing these boxes. I can't tell you how awful it was. It was so difficult. But what I loved about it was the sense of community, that sense of camaraderie that I got from the shop floor. And that has stayed with me my whole life. I left here and went to Israel. I became a dog trainer. Actually, before I became, and this is one of my favorite dogs. It's a Rhodesian Ridgeback, African dog, South African dog. And here I'm teaching it not to eat the chicken on this particular occasion. But before you work your way up to being a dog trainer, you start off as a glorified pooper scooper. 70 dogs is a lot of poop, and my job was to clear it up day in, day out. From that job, I work your way up to the dog trainer's dummy. And they give you this large leather suit. It goes from your ankles up to your neck. It's about 7 millimeters thick. And you feel quite protected by this suit. You have a large leather arm with a metal bar in it and a Hessian sacking around it and a big stick. And that's where your training starts. And it is terrifying. The first day that they would have a German Shepherd or a Doberman or a Rottweiler at one end, and they get to come out of the cage for 1 hour a day. And in that 1 hour, it's okay for them to go and eat someone. And they let this dog go, and it hurtles down the dog guard. And the first day I was out of there, I turned around and ran, and the dog thought, great, a moving target. And it chased me out of the dog guard. It hit me on the back. And it was a real whirlwind of a fight. Anyway, 2 or 3 days later, I got my own back on the dog. It's the Uzi 9mm semi-automatic machine gun training. And of course, the dogs have to get used to being shot at. And they let the dog go, and I'm ready, arms ready, and you're shooting. And the dog's running towards you, and it's dodging the bullets, and it's... Anyway, the last bit's not true about the dodging the bullets, but it is the most terrifying experience that I had, until I got into science, that is. I left dog training to go to Australia. And I got a job working on the Flying Doctor fights. What the plastic moulding firm had taught me is that I could make things. This is us now flying into one of the ranches about 1,000 kilometers out into the bush. And we're just about to land on this makeshift runway here. Some days we'd be flying over the bush, and there was one particular day when the light aircraft just dropped out of the air. And as it dropped, everybody's heads hit the roof, and the pilot points down, I'm at the front, and he points down to the bushfires. And we were bouncing on the thermals of these huge bushfires. It was spectacular. We came into land here, and my job was to make a piece of X-ray equipment that could be transported in the Land Cruiser out to the Aboriginal settlements. This is my piece of equipment that I made. And we would then set it up in one of these shacks, and I would take chest X-rays of the Aboriginal community to work out the amount of tuberculosis in those communities. It was wonderful. There's the sense of adventure I just loved about it. But it was also my first taste of research, and more than anything, I knew I wanted to do more of this. I left Australia and came back and did some night school or some diploma courses. But in that time, I met a group of people that were interested in climbing. And I was a bit unsure about showing this. It's like some 1980s boy band. And I'm still a bit unsure about it anyway. These are the guys I trained with, and we did 5 summers in the Dolomites. We just spent 2 days on this face here. And it's like doing a PhD in some respects. You're climbing things that other people have done, you're cutting your teeth, you're learning lots of new skills along the way. For that one day, you're going to try and climb something that no one else did. And that day started to happen as I moved out from the Dolomites. We went to Africa, and we'd spend, this is the north face of Mount Kenya, we'd spend months at a time trying to climb these new routes that no one else had climbed before. I moved from Africa and then went to live in South America for a couple of years in order to try to do this. There is a most special day you can have. You know, to start with, it is a bit like a PhD. You're slogging up these slopes endlessly and you're wondering why you're doing it. But there's a moment when you stand on part of this earth that nobody else has ever stood on. You look out at a view that nobody else has ever seen. And I believe that is one of the most special moments that you can have in your life, which makes for me why climbing is so similar to science. I left South America and came back and got into a PhD. And I met a guy called Jim Al-Khalili. And we were approached by Channel 4 to go in search of Einstein's brain. There was a rumor it still existed. And I'm going to show you this because it was one of my best trips. This is me and Jim having a debate about Einstein's brain. It was a great job, this.
Southern California, hunting for Einstein's brain.
Let's do it.
Mark is sure Einstein's brain will be insight into Einstein's genius, but Jim is not convinced.
Einstein's genius is his ideas. You never find ideas in the jar somewhere. I mean, he revolutionized physics in the century. From his cycle of relativity came everything, Big Bang, expansion of the universe, black holes. All that is entry, the creativity, the genius, the intellect, every mind, every brain, every conscious thought. Those don't exist anymore. Conscious thoughts don't exist anymore. But what created the conscious thought? Whether it was certain areas of the brain, whether it was the synaptic connections, they were products of the brain. And certainly parts of those are still around. I'm not expecting to find everything, but I might be surprised.
For physicist Jim Lythgoe, imagination is so extraordinary it can produce a theory as wild and abstract as relativity cannot be explained away by mere biology.
I'm going to show you another clip. It's one of my faves that we did from for Channel 4. It was when we were in the lab and they approached us with this project called Easter Egg Live. And they were looking at different animals, from spiders to chicks, and how they hatched out. And they wanted to open the show with a chick hatching out of an egg. And we did this by having incubators in the lab and putting them into the MRI scanners. So this is the chick in the egg. This is the air sac at the top. And we take lots of slices through the developing chick. And then from that, we were able to reconstruct that data. So this is quite a young chick, and you can see the back's not in contact with the shell. And then as the chick gets older, it fills the shell up, and it starts to put its back against the outside of the shell. And just before they hatch out, you can see the eggs rocking as it's moving around. It then needs to breathe, and it breaks into the air sac here, and then it breathes there for a while. Eventually, it runs out of air. And it's that lack of oxygen that switches on certain genes and the drive to break through the shell using this tooth, which is on the top of its beak. And I think to some degree this emphasizes why imaging is so powerful. It's so difficult to conceptualize what is going on inside that egg until you actually are able to visualize it. Something that I'm deeply proud of is the Cheltenham Science Festival. It's been running now for 20 years, and 20 years ago we started this festival, which was quite small at the time, and it has been on quite a journey in this last 20 years. So what's Cheltenham Science Festival like? It is a festival of science. The first year we ran it, there was only a couple of thousand people that came. It was relatively small, a couple of small venues. Now we have 12 different auditoriums, big top tents with 1,000-seater. We have about 70,000 visits. It's the first week of June every year. We run a competition for the New Voices of Science, which is run in 22 different countries. In fact, when it was run in Egypt, 25% of the population watched the final on television. We installed the first MRI scanner at a festival and ran a national competition for the kids to put things into the scanner. And of course, the fruit comes first, and the apples and the bananas. And then they got more adventurous from someone brought in a snout and a pair of ears. And finally, a pair of testicles, which we have, of course, never scanned before. The wonders of science, that's what we like to bring alive through the festival. So why have I told you all this? It's because I believe that my slightly unconventional, circuitous route into science has informed the type of scientist that I am, and it informs the type of science that I do. I'm now going to have a slightly uncomfortable segue into the lab, which is Cabby. We opened about 10 years ago, and tonight I just want to share with you some of my, perhaps not the most impactful pieces of research, some of the pieces of research which are my favourites, some of the ones that I just really love, some of the wacky ones, and some to give you a little food for thought, perhaps on the way home. I'm going to try and get through our journeys through trying to find a hole in the heart, magnetic navigation in the brain, the invisible body. Could we make someone transparent? The next generation of imaging technologies. And finally, I'm going to finish with some brain imaging. I'm going to start with one of the challenges that we were given. It was the first project in the lab. And it was given to us by Pete Scamler at the Great Ormond Street Hospital. And he was looking for the genes that were responsible for hole in the heart in babies, blue baby syndrome. And he challenged us with making a technique to find the holes in the heart. This, at first glance, is an embryo. It's not a fetus, it's a mouse embryo. It's about the size of your thumbnail. The heart is about the size of a pinhead. And we were looking for holes in the heart that were the size of a human hair. The idea is that you knock out lots of genes, and then you try to find the one embryo that has the hole in the heart, then you understand which gene is responsible for that particular hole. Once we had a picture of the heart, bearing in mind that this is the size of a pinhead, we're able to look at the structure of it. And here we can see this is the left ventricle, the big pumping bit of the heart. But instead of the blood coming out and coming down the major vessel and out to the body, it gets pumped from the left side directly into the right through a little hole just here. And this is the hole that causes the blue baby syndrome. Of course, once we've found the hole, then we can look at which particular gene was affected. Once you understand that, then you can look at diagnostic tests, prenatal diagnosis, and to see if there would be a likelihood of a baby having that particular defect. Our next challenge was a moving target to image the beating heart, but not the beating heart of a human. Our hearts beat one beat every second. Actually, mine's going a bit faster than that, but it's going one beat every second. A mouse heart beats 600 beats a minute, 10 times faster than humans. So one heartbeat is in the 10th of a second, literally a blink of an eye. And in order to get a cine picture, we have to take 20 pictures in the blink of an eye. Now, this is a mouse heart, and I've slowed this video down so it's the same rate as a human heart. So this is a mouse heart the size of a pea beating at the rate of a human heart. It's twice as fast now, twice as fast as the human heart, 5 times, and then the actual rate of the mouse heart beating. And of course, once we have the cine picture, then we can start to slow it down. We can start to look for the function of the heart. We can start to have a look at when the heart has an infarction. And then, once we have that, then we can start doing something to try to repair it. And Daniel Stuckey in the lab is putting patches onto this organ that beats so quickly to try to regenerate the heart once it's had a myocardial infarction or a heart attack. Now I'm going to have a real sideways step from imaging. This is quite a blue sky project. Over the years, we've been asked to track cells. There are cells to regenerate your brain, heart, kidneys. And people want, when they're injected, people want to know where those cells go. So they asked us to develop techniques for finding out where these cells go. But it was clear when we tracked the cells, actually not many of the cells went to the heart or the brain or the kidney. And we came up with an idea to try to increase the amount of cells that went to that diseased or damaged organ. It started as a cardiovascular project, a vessel project with people with damaged vessels in the heart. could we attract cells that would regenerate vessels using a new technology? So what we did was to put tiny nanomagnets into the cells and then put a magnet on the outside of the heart or a blood vessel to try to attract the cells to the sites of damage. Now, of course, you know, we all generate these ideas in the pub late at night, and they always sound great. So we rushed back to the lab the next day, and we wrote it up for a grant. And we wrote this up, you know, about what we were going to do with the magnets and the nanomagnets and how it was all going to work. And then comes back the reviewers' comments. And the reviewers wrote back, the physicists, that is me, on this project do not understand the fundamental laws of magnetism. This project will never work. Which was pretty damning, to say the least. Anyway, we did set out on this project, and this is why this next image gives me a lot of pleasure if you're in the audience. This is, oh, let's just go back. Let's start that again. So this is a droplet of water, and it's under a microscope, and each of these black dots is a cell, and it's black because it's got our nanomagnets in. And then all we did was to put a magnet on the outside of the water droplet. I can't tell you how much pleasure I get out of that single image. Once we'd done that, I thought, this project is going to work. There is no doubt about it. From that, we got very excited. And we tested it out in animals, we generated arteries, we got all these cells there. So it was, as it was one of our first projects, we decided to do a press release. I'd never done a press release before. So I wrote in the title, stem cells, which they were, containing nanoparticles and nanomagnets could be guided by magnets, which I thought was fair. And on Monday morning, the papers got hold of this, and I think it was the Times or the Telegraph, I can't remember. It's the Telegraph. Magnets could be used to heal hearts. Okay, it's a cardiovascular project, that's fair. Tuesday, the Times. Tiny magnets that help heal injuries could be used to treat cancer. Friday, the Daily Mail had got this. You know what's coming, don't you? How magnets may help to heal everything. Do you know how many people phone you up when you can cure everything? It's overwhelming. We had our fingers slightly burnt with that one. We've taken this one step further. What are we doing with this technology now? And of course, we use the big MRI scanners, and the MRI scanners are magnets. The question is, could we use an MRI scanner to start to guide biological material around, cells inside the body? And this project's called the Minima Project. And quite simply, we place one of our nanomagnets here. They're called seeds, about the size of a grain of rice. We can then pick them up with the magnetic field of the MRI scanner and then guide them, and in this case, this is a brain tumor, to the location. And then we fire a radio wave, which then heats up these thermoseeds, which then kills the tissue, ablates the tissue. We then can move it around the tumor, ablating all the tumor as we go round, and then move this back out to the point of insertion. What I would love to be able to do is to take the MRI scanner, a diagnostic device, and then change that into a therapeutic device with just a small modification. That is our overall aim, to make all these devices theranostics, a combination of therapy and diagnostics. Okay, now, the Holy Grail. Wouldn't it be amazing if you could make someone transparent? You don't need to put a jellyfish in an MRI scanner to be able to see what's going on inside, because it's transparent. Which sort of started this conversation of what is transparency, something for you to dwell on the way home. Why are some things transparent? And this started to think, could we make organs transparent? And what would we do if we were able to make an organ transparent? We started with hearts. This is a heart that's sliced in half. You can see the pinkish reddish color of the heart. This is a heart once we've made it transparent. You can put it on a book and read the book through the transparent tissue. And once you have this, then you can start, as with the jellyfish, start getting fluorescent light. You can label it, make things colored with fluorescent light and get properties out, images out that are just not possible with any other technique. Okay, this is the bit that I need you to put your glasses on. Has everybody got a pair of glasses? If you haven't got a pair of glasses, just shout out and I can drop a pair too because I've got lots at the front. Has everybody got pairs of glasses? So pop your glasses on. All right, Robin, have we got there? It takes quite a while to get them out of the packet. Our COVID-friendly glasses. What we're going to do is I'm going to show you everybody got them on? Right, we can drop the lights down now. I'm going to show you a picture of a tiny tumour. It's only 2 or 3 millimeters across. And instead of sitting on the outside of the tumour, I'm going to take you through the tumour. In order to get your eye in, when I show you to start the picture, I want you to focus on the front bit to get the 3D effect. So we'll try, it'll take a couple of seconds to get your eye in. So I'll start the video. So just let your eyes relax and start to focus on the front bit. And then it will start to stretch out slowly from the front to the back, to the extent that you can start to sort of put your hand up and it's as though you can touch it once you've got the full 3D. With this, of course, we can walk inside the tumour, but you'll also see there's colours attached to it. We can colour code the blood vessels with different sizes. We can have a look at the distribution of how leaky the blood vessels are. We can look at how far the drugs go. But importantly, we can see where the drugs don't go. And we can see whether certain blood vessels are stopping that drug to get to particular parts of the tumour that the therapy isn't working on. Can you give me a sort of a nod if you got that? Sometimes it's quite difficult in different, yes. Oh, excellent, right. You can take them off now. Now, I appreciate online that you might not have had your squiggly glasses at hand. So I'm going to show a different one that works in normal 2D, you'll be glad to hear. This is one of my favourites. Instead of making a small tumour transparent, this is a whole organ. It's the brain and the spinal cord. Again, we sit on the outside of tissues. We sit on the outside of the brain, but imagine we could colour every single brain cell. And you could wander inside the brain and look at every single connection, memory, emotion, sensory motor. And then you could take a journey like a roller coaster, an escalator going down through the spinal cord. To look at the reasons perhaps why we have peripheral pain, or to look at spinal cord regeneration. And that was unimaginable for me 10 years ago. So at the moment, we've done small tissues and then slightly larger tissues. And now I want you to bring this back to clinical and human studies. And this is a lovely project that's been done by Claire Walsh. And Claire goes to Grenoble in France to try to image a whole human lung, but image it from the whole lung right down to the individual cells, just enormous amounts of data. She works with Paul, and this is the lung inside their setup before they put it into the world's largest microscope, the brightest light source, the European synchrotron radiation facility. Here you see the sort of X-ray of the lung. But from that, we can go down and down inside. You can sort of recognize the branching patterns. But as we start to pull into the cellular level, the black spots are where the air is. And a lung should be full of black areas in order that we can breathe. Now bear in mind, this is from a COVID patient. And you can see all the tiny little air sacs, the ones that are still useful. But the rest of it is full of cells and fluid, which is why we have such difficulty with the breathing during this particular virus. Now, around the air sacs, you can see little white lines. These are the individual blood vessels. And within the individual blood vessels, you're starting to see individual cells. And using this kind of info, you can see these cells here, the individual blood cells sitting around the air sac. And it's the communication from the air sac to the blood cell which decides how much oxygen we will have in our body. And poor communication, which is what we're trying to understand, is why the patients end up on intensive care and unable to breathe. So, to take you on to the next generation, where are we going with all this? All of you with a keen eye, and I know all of you have spotted this, that was not live imaging. The question is, can we do all of that, but in live patients? And I want to introduce you to photoacoustic imaging. And we're going to turn to nature for this. This is a bolt of lightning, a lightning strike. When lightning strikes, the lightning bolt comes down and it heats up the air. And it heats it up really quickly and you get a thermal expansion. It gets incredibly hot. And that heating up causes the thunder. The lightning and thunder is this exchange of energy from light into sound. And that is what we use for our new technology. We fire a laser into the body and then we use the body to convert that into sound waves. So how does this work? I'm going to do a little demo. Might need to bring the lights up just here. What I need is either a credit card or a card. This is the tricky bit. There's never usually a rush to give me credit cards. I can see you haven't given me a credit card. That's how much you trust me. It's one of these. That's your backup. Okay, that's my backup one. If it doesn't work, I can see you really trust me. I'll hide that back here for the moment, just in case. All right. So the idea is I have a flashlight. That's our lightning. I'm going to shine that onto the credit card. The light will go into the credit card. It's absorbed by the pigments. And because it's given in a nanosecond flash, the credit card should expand. And it's like a whip crack. Will give out a tiny sound wave. I'll convert light into sound. And you are going to hear for the first time the sound of a Royal Society Medicine membership card from Roger. This is our world's first. Right, it's a... let me make sure I've got everything on. Okay, so you have to be quiet, all right? It's quite a quiet sound, all right? So I'm going to put this onto... I've got a very sensitive microphone. I'm going to rest this on. Oh, goodness. Right. And now I'm going to flash this onto the card, and we will listen. Is that mic on? Fingers crossed. Hopefully the mic is on. Oh, God, yes, brilliant idea. So, yeah, okay. So even with the speakers and the mic, fingers crossed, absolutely quiet. Okay. 3, 2, 1. The sound of a credit card. Can I do it again? I just love this so much. I can't tell you how much I love this demo. I get so much pleasure. What, it's worked. Roger, you're a star. Never do it with... Okay. 3, 2, 1. Oh. Whoa, it's amazing. The sound of Roger's card. Thank you, Roger. Thank you. Let me switch this off. So, okay, there's one thing doing it on a table. Let's switch that off. One thing doing it on a table, and there's another thing doing it inside the body. So, in the lab, we have a large laser, a near-infrared laser, which is just like our flash. The light gets into the body, and near-infrared light penetrates deep into the body. It's given in a nanosecond pulse, a tiny blip of light, and as it hits red blood cells, which have pigments in them, the pigments absorb the light and they expand and they give out a tiny sound wave. And we use a very sensitive microphone around the body to listen to the sounds that are given out from the red blood cells to create pictures of the blood vessels. Can we drop the lights again? And let me just run this. So the picture that you're seeing here is created from the blood vessels and a small tumour given out from tiny sound waves that are emitted from the individual red blood cells. But we've also been able to create, and this was a first, a way of putting an agent, a dye, into the tumour such that it also gave out a tiny sound wave. And with different colours, using different pigments, just like painting, we get different effects. And we can tune the laser to the red blood cells or to the tumour, and then we can overlay one on top of the other. We can then give drugs, vascular, disrupting agents, which destroy the vasculature. And you can see them curling up as they stop supplying the tumour in real time, over time. And that is not possible with any other technique. How are we using this clinically? This is a lovely image from Adrien Desjardins. And this is a picture of the blood supply to twins using photoacoustic imaging. And one of the problems with twins sometimes is they don't have an even distribution of blood flow, and the surgeon has to go in and redistribute the flow so that it's even between the twins. It's an invasive procedure to even find out which vessels to manipulate and alter the junctions of. So here we're seeing the photoacoustic imaging giving us an intricate, detailed view of the blood supply to the two twins that then they can use to redirect that blood supply to even it up. Okay, the final part of this lecture is on a brand new brain pathway. And hopefully it's something to think about on the way home. I'm going to talk about the brain's waste removal system. It's in every textbook. Your brain weighs 2% of your body weight, but needs 20% of our energy in order to function. And as with any, engine, high-performance engine, there are a lot of waste products. And waste products form within the brain. But also, the brain accumulates toxic waste products, perhaps in the form of plaques. And it's these plaques that lead on to Alzheimer's disease. The question has been for such a long time, for decades, what is the brain's waste disposal system and how does it work? How does the brain clear itself? And why doesn't it clear out these toxic proteins? So this is the current view of sort of the water in the brain. In the center of our brain, we have these fluid-filled sacs called ventricles. About half a litre of fluid is produced a day up in these top bits here. It's then shunted around through all these cavities right out to the bottom and then out around the outside of the brain. And it just goes round and round down the spinal cord. Importantly, it has not been suggested that the fluid communicates, exchanges, or goes into the brain itself. It doesn't contribute. to the washing of the brain. This is our traditional view. We have the fluid around the outside here. We have the fluid-filled sacs in the middle, and these are all the blood vessels. But I want to take a closer look here as we go down the blood vessels deep into the brain. Because what we believe now is that there is a brain pathway, a new brain pathway that has been missed. And this pathway is a brain clearance system called the glymphatic system. The idea is that instead of the fluid just going round and round the outside, that it tracks down minute canals next to the vessels. And then when it gets to the end, it comes out of those vessels, goes into the tissue, and starts to wash out the waste products. If I take a magnified view, this is one of the vessels, and the green is the fluid from the outside of the brain. And the simple notion is that this fluid will move through the cells, our neurons, and wash out all the waste products. And it goes out down the veins here. The first question is, it's fine on paper, but does that pathway really exist? So we went to our imaging and we inject a small amount of dye into the fluid-filled spaces around the outside. This dye shows up on our MRI scanner. And in principle, if the textbooks are right, it should just stay around the outside of the brain. It should just go round and round. This is a slice through the brain. We want to see if it's gone into the brain tissue. And this is the video running as we follow it over time after the injection. In a very short period of time, we're starting to see the coloured areas as the dye goes directly into the brain. 30 minutes is incredibly dynamic, this process that happens, this brainwashing as it goes from the outside into the tissues and out again. So this is what it's like, as I've just showed you, with the tap on. The tap on, the fluid's coming through, it's washing the brain. The question has been, for us, what happens in Alzheimer's disease? Is this tap been switched off and therefore we can't clear the toxic proteins and you get a build-up of the plaques and then Alzheimer's disease continues? So this is Alzheimer's disease. And you can see the limited amount, the lack of colour, the lack of dye that's gone into the brain. It's as though the tap has been switched off. The question is now is, if we could open the tap up again, would that provide a cure or be able to prevent Alzheimer's disease? Now, of course, what you all want to know is, what is it? How do we turn the tap on? And if I was to tell you that, I'd be stealing Ian and Doug's thunder for the next year, as they are working on both what's called an inhibitor and an activator of this pathway. But what can I tell you for you to dwell on this evening about how we wash or clear the brain? Okay, firstly, it's not about going to get a cuddly pet and just spending the evening with it. If it was only as simple as that. It is, of course, sleep. This pathway seems to be activated during sleep. Why do we sleep? Has this been a missed function of sleeping, that this pathway becomes activated and allows the clearance? Bendy animals. It's not bendy animals, but of course it is. Exercise. Exercise, and that makes sense that we already know that exercise is good for the body, but perhaps emphasizing that it's also good for the brain and the mind as well, in a different way than we thought about. And finally, this may reinforce your views, or it might not cheer you up, but it is, and there are no free lunches, as we know with the brain, It is, of course, alcohol. High levels of alcohol seem to reduce the brain clearance. However, and there's a big but here, low levels seem to increase it, which will be something for you to dwell on the way home. I know what you're going to ask me. How much sleep, how much exercise, and how much alcohol? Probably, if you're asking me that question, you probably need either more sleep, more exercise, or less alcohol is probably my feeling, so I'm just preempting that question. On that note, of something for you to think about, I want to thank a few people. 4 people that are here tonight from the lab. Daniel Stuckey, who is just amazing, and all those cardiovascular images and the patch and regenerating the heart. This is all Daniel and his team. Jack Wells, who leads all the neuroimaging, all the amazing techniques for looking at the brain. Tammy Calber, that does all things cellular and molecular, all that tracking of the cells. And Ian Harrison, who is our brain clearance expert, and will be revealing, hopefully, how we get an activator. The rest of the lab, lots of who are here this evening, thank you so much for coming along. All the alumni and the endless list of collaborators. It just leaves me to say thank you for this award. I can't tell you how much it means to me when I look back to the factory such a long time ago now. It's been a hell of a journey, to be quite honest. And I wouldn't recommend it to anyone, especially the dog training. I'd like to thank the Royal Society of Medicine, who have just been so incredibly welcoming and warm to me. I would like to thank the family for all the amazing adventures that we've had. And I would also like to thank every single one of you for coming tonight, and all the people online for listening. Thank you so much.
Mark, let's stand in the middle there, Mark. Wow, that was amazing, wasn't it? That was fantastic communication, absolutely amazing. We've talked about the heart, we've talked about cancer, we've talked about the brain, we've talked about everything really in medicine and the amazing advances that imaging in my specialty. I'm just a humble prostate surgeon, but I tell you, MRI scanning of the prostate, enabling us to tell where the tumor is, has completely revolutionized prostate care. So we have an opportunity to answer a few questions. We've got some online here, but anybody in the audience, normally we pass a microphone around, but with COVID and so on, we think it's not safe to do that. So you're going to have to speak in a very loud voice. So anybody in the audience? The lady in the pink shirt, introduce yourself and speak very loudly. Ask the question. I'm going to repeat it into the microphone so that the people online can hear. So, who are you and what's your question?
Hi, I'm Alex. Can you hear me?
Yes, Alex.
Perfect. So my question is, you know the rice brain device for early of the brain tumour? Is there, are you seeing that as being translated into cardiac activation replace traditional catheter-based methods?
Okay, so
Mark, repeat that question because they won't have heard it online.
So the little magnetic seeds that we can guide through the brain, could that be used in the heart? And they use catheters with little tips on them for ablating parts of the heart. Could it be used for that? So where it has been used in the heart, there is a company that's been set up, and I won't mention their name because it'll sound like I'm publicising them, although I have nothing to do with them, where they have taken this idea and they have 2 very large electromagnets in theatre. And the catheters, they put a little magnetic tip on them. And they use the electromagnets to guide the catheters through the vasculature. So that's where it's been used. The reason why it hasn't been used anywhere else is that we're the first people to be able to do it, is to be able to put them into a device where you can image it, and then also to be able to do the heating at the same time. And actually where we started was with Mark Emberton and prostate work.
Yeah, good man.
And so prostate is probably where we'll try first with Mark, and then on to the the brain next, and we have a series of collaborators. So it's in its early stage. The question, could it be used in the heart? So again, if you put it onto the catheter and use the catheter and then fire the radio wave in, it will heat up and ablate. There is no reason why that couldn't be done. And the real advantage is that you get the navigation on the way through, but importantly, it's the imaging that goes alongside it. How do you know where your little thermoseed is if you don't have some imaging device to tell you where to go? The ultimate blue sky would be you get the patient and you do an MRI scan and you plan out a particular area of the heart or a particular area of the brain that you want to go to. And then you could set up an automatic process where you image it, and then the image then tells the scanner how much to move it, and then you image and move, and it goes on some predefined track until it arrives at its final point. You then ablate, and then you can decide how far to go, and then it would remove itself down that track. So in principle, the whole thing could become completely autonomous. In fact, I'm hoping that in the next, I'm going to curse the paper now, that it will get published in, look, Rebecca's smiling at me, because I'm going to curse the paper now, aren't I? And hopefully in not too long, and then we will do a little bit of press around it.
Mark, I was intrigued by your history of working in the factory, dogs and all that stuff.
Oh, yes.
What was there a moment, a sort of Pauline conversion, where you suddenly decided that you had to do some work? I mean, did you fail all your exams because you didn't bother to do any work, or just tell us a bit about that? What changed your life?
Oh my goodness, there's lots within that. The sense of failure was quite profound at that time. Mm-hmm. I remember my dad was so disappointed. My dad's watching tonight. Hi, Dad. I've always wanted to say that. I remember my dad being really disappointed, and that sense of failure, lingered with me for a long time. Why didn't I do particularly well? I just did not engage with the system. I don't think I'd had anyone that really inspired me, that told me if I did this would happen, and so many wonderful things. So when you haven't got any qualifications, there's a limited amount you can do. But I did want a sense of adventure, which is why I went abroad. It was when I went abroad that I met people that had been to university, and they told me that they're all going back home to these amazing jobs, and I'm going back to a factory. And I just thought, I've got to find a way back into the system. And it was not easy. It was so difficult to convince people that you are capable of doing this stuff when you've got no history whatsoever in it. The existing systems that we set up are set up there because it's easy to trust someone that's come through an educational system. In fact, there's a lot of snobbery still around the educational system. Certain universities, there's real credit if you've been to one of those, because there's a certain degree of understanding of that system and trust. Therefore, when someone comes from the outside, it's very difficult to know what they are going to be like, and I completely understand that, and because I went on the journey through it, that's why I'm saying I wouldn't recommend it.
Maybe, let me interrupt, maybe the fact that you haven't been kind of brainwashed like the rest of us through the university system, that's what makes you so brilliant with your ideas, which are so kind of zany, and would you think there's
I did say that, I reflected on that in the middle, saying that I think all that malarkey has informed the type of scientist that I am. I can't believe it hasn't informed the type of person that I am, the type of science that we do, the risks that we would take. I love the risk-taking, crazy, highly experimental projects. I just love that. And I think that's what takes me on that journey to tackle some of those real challenges. And of course, a lot of them just don't work, and those are the risks that you must take, and you've got to have strategies around that. So it's definitely the risk-taking has been an element for me, and I'm sure that has pulled through from the climbing.
Okay, there's some questions coming online.
Oh, no. God.
Melita Irving is taking over from Robin as chairman of the Ellison Cliffe Foundation. So good for you, Melita. She says, thanks to James Bond for the inspiration behind this question, but can nanoparticle technology target DNA, for example, to treat genetic disease or cancer? And she apologises for not being there. She's busy tonight. So that's a... There's quite actually another question too about whether you can correlate biopsies with the imaging that you have. Is that going to be certainly that's the case in the prostate, isn't it? You can see the tumor, but you've got to biopsy it to get the tissue. But I mean, is that true of the other issues you're looking at, the heart and the brain? Do you think?
So yeah, so still, we're at this, if you want to find out what's going on in the tissue, the old and traditional and trusted method is that you stick a needle in, take that piece of tissue, and then you can do tests on it. Everything that we've tried to do in the lab has been to try to find that information, but without doing the biopsies, to try to use imaging, instead of just looking for the structure or the anatomy of an organ, trying to understand the microstructure, trying to understand things down to the individual cells. We can even follow genes. You can switch genes on in the same way as the jellyfish does with the green fluorescent protein. You can switch genes on that produce a dye that will switch on our photoacoustic system. So you can follow those genes as they change over time. In fact, we have a project now that we're trying to see why brain tumours, how they migrate in the brain. And it looks like the brain tumour starts quite small and the centre quite dense, and then cells set off. They migrate, they take on new functions as they move across the brain. So as they move across the brain, normally what you do is you take lots of samples out to try to work out what these cells are doing, where they're going. But we're trying to then get these cells to switch on a gene that says, I'm this particular cell, I'm here, and this is my new function, which might be to push through tissues, to go through grey matter or white matter, to start to divide and multiply here. And then that will show up on the photoacoustic scanner. So everything that we can do with histology, we're trying to move to non-invasive imaging.
Yeah. Well, I'm going to ask you a question now. Any other questions in the audience? Put your hands up.
I'll take anything.
Anything. Okay,
oh my
sir,
goodness, no, I shouldn't have said
the man
that.
with the blue shirt and the glasses.
Yes.
Hi, I'm Ron Knight. What would be your imaging technique of preference for looking at blood flowing through medical devices to enable you to design them better?
So if it was a...
Just repeat the question.
So it's, which imaging technique will we have to look at blood or something flowing through a medical device? It depends on the size of the tubing in the device and where it is. But MRI is wonderful for looking at flow through tubing, even small vessels. You can use a contrast agent. And CT, if you just type in CT scanning and blood vessels, it is amazingly detailed. The problem with that is you've got X-rays, radiation doses, but with MRI, it's completely non-invasive. So if they were attached to patients and things, the MRI route is great. But the CT, amazing detail, MRI, completely non-invasive. But they're good for flow, and you get quantitation out now. And there was a question here and a question here. I'll go here and, oh.
Yeah.
Oh, yeah.
Sir.
The last subject talked about Alzheimer's disease. That's very important, I would have thought. The brainwashing process. Is that, you mentioned that sleep is one of the most important factors. So is there a suggestion that people have more sleep and they can be artificially good for sleep so that the brain washes through its toxic proteins and those with Alzheimer's, is there a possibility of actually?
So, big questions. So the sleep is one of the first studies that came out. And it's really difficult to investigate. So there's a caveat there. So the way I'll come back to the sleep, the way we've approached it, instead of saying, right, get more sleep, which is actually the worst thing you want to tell someone sometimes is get more sleep if they're struggling to sleep, because that's immediately going to stop you sleeping because you're starting to think about it. But is on each of the blood vessels, there's like a little tap that you can turn on and turn off. And our aim at the moment is to look at this tap to see if we can increase the flow through the brain, to increase it like that. And we believe that's probably an easier way than getting people to sleep. What is clear is that people with Alzheimer's struggle with their sleep. The question has been, do you struggle with your sleep first and then that might lead on to Alzheimer's? Or is it the other way around, that it's people with Alzheimer's just struggle with the sleep, and that's a byproduct of it? That's not known at this moment in time. And it's really difficult to disentangle those two. That's why for us, the data that we've got suggests that if we can increase the flow, we know if we decrease the flow, we can't get rid of these proteins. The question is, can we just increase it by putting a little tap on it? There is hardly anything out there at this moment in time, and this is a brand new pathway, and everybody is still scrambling to get their head around it and the mechanisms by activating that.
Mark, we're running out of time.
I've got one question here,
okay.
Right? And then one question here.
Thank you. My name is Sophia.
Hi.
Hi. You talked about therapeutic and the
Yeah.
concept of therapy and diagnostics, and you talked about the transparent tissues or organs,
Yeah.
and maybe that as a conduit to seeing if drugs work or where they're not working.
Yeah.
So I'm an ophthalmologist, and we often say that the eye is the window to the soul. So I'm just wondering, as a proof of concept, do you think theranostics for the eye and seeing where drugs are delivered and where they don't work? For example, in the retina, we do have good therapeutics, but we can't often image exactly where or where not they're working. So do you think the work that you can do could translate to an organ where you can directly visualize what's happening?
So you guys have it easy. You've got this beautiful little window that you can just look in. It's like having a transparent bit of the body. And there are so many techniques that look at all the amazing blood vessels at the moment. We all struggle to see where the drugs go. So the direction that people are going is to understand the networks, to be able to map out the networks incredibly well, and then use artificial intelligence to understand the drug and then predict where that drug would go. So instead of trying to image the drug, you would look at someone's blood vessels, and then from that, you would predict where that drug went, and then tailor the therapy before they started based on that. Because it's so difficult to track the drugs that my guess is that the modelling and the simulation will come first, and the AI, of which there is so much now in ophthalmology. So that's my guess before we'd be able to look at the distribution of the drugs.
Last question.
Iski!
You've been talking a lot about the MRI scanner, so I was wondering how it works.
Oh my God, no!
That's a tricky one. Yeah.
Oh my God, it's my son. Iski asked me, I've been talking a lot about the MRI scanner, and how does it work? I struggle between, I'm either thinking, well, I haven't got much time, let's chat about it at home. I'm thinking, right, I'm going to have a go at this. Okay, so I'm going to have a go. Are you ready? So in the body, it's full of water. Okay, we've got waters everywhere. We're 70-80% water. And all the water molecules wander around inside the body. But then when you put the water molecules inside the MRI scanner, they all line up with the MRI scanner. All right? And then when they're in there, you fire a radio wave at them, and you knock them all over. And then you let them go. And they roll back up to the original position. And as they're rolling back up, they give out a tiny radio wave. So the pictures that we see are created from tiny radio waves that are emitted from the water molecules in the body. Okay. Oh my God, pressure. There's just so much pressure.
Well done. We benefit. I know this. All your children want to ask you questions. I was going to ask you, what are you most proud of? But I can see what you're most proud of. Amazing. Apart from all these amazing things, is a family of 4 there, and good old mum who probably takes care of them. So on that note, I'm going to hand over to Robin, who is chairman of the Ellison Cliffe Foundation. There's an opportunity for ask more questions and drinks outside in the atrium. So after Robin, we can adjourn straight outside. Robin, over to you.
President, thank you, ladies and gentlemen. Well, we've had a fascinating travelogue, travel through the continents, travel through an unconventional but brilliant career, and of course, travel through the human body. One or two of the highlights for me were the 3D film. We had the glasses, but not the popcorn. and the idea of the brainwashing, simply stunning. These lectures were founded by Caris Ellison and her husband, Percy Cliffe, and the whole idea of them was to explore the interface between science and medicine. And my goodness, that's been done tonight. They would have been thrilled had they been here. Caris died at the age of 98, 2 years ago, and Percy predeceased her by some 20 years. She was a psychiatrist, he was a medical physicist, so they explored that interface intimately themselves. It's quite scary talking not just to a live audience, but also remotely to a few hundred out there. I've reached the stage in life where I read the oldie. And in this month's issue, there's a story that Giles Brandreth gives of Judi Dench and Ian McKellen when they were both very young at the start of their theatre career. And Judi Dench was absolutely terrified before going on to stage. And she said, what I've decided to do, she said, is talk to the 3 chairs in the front row and think it's the Father, the Son, and the Holy Ghost. To which Ian McKellen said, surely, dear, they were sitting in the one chair. We have indeed had a tour de force. Mark Lythgoe has awards in neuroscience, imaging science, and it's easy to see why he also has an award in communication science. It is not, we've had Nobel Prize winners here in the past at the RSM who aren't always as clear or as skilled at presenting their ideas as tonight's lecturer has been. So I do believe that Caris would be really thrilled if she had been here to listen. Now, we have a medal to give you, sir, with the images of Caris and her husband on one side and the crest of the RSM on the other. So can I give that to you first of all?
Thank you so much.
You can have, I've given your wife a sneak preview, so if it comes out like this.
Sorry, God, it's like Christmas.
I know. So then you can open the box and you can see your medal.
Oh, wow.
And you know it's yours because it's got your name on.
Thank you.
There is an honorarium which goes with this lecture. I asked Charlotte, who organizes the lectures, whether we had the check. She said no, that was very old-fashioned, so she's going to get your bank's details and do a transfer. And President, now I think it's time for us to titrate our alcohol intake carefully to clear our brain of all those noxious chemicals. Thank you very much, Mark.
Thank you. Thank you for your time.